All right. Thank you very much. Good afternoon. It's really a pleasure to have Dr. Angelica Thomaz Vieira here as a speaker today. And Dr. Angelica is a biologist with a master and doctorate degrees from the Federal University of Minas Gerais, with really great international experience that took her to Australia, to the Garvan Medical Research Institute, and postdoctoral periods in Strasbourg. And she was a member of the Brazilian Academy of Science, an affiliate member of the Brazilian Academy of Science, which is really a great honor and well-deserved, and also received really important prizes, like the Young Woman in Science and the Teresa Kipnis prizes from the Brazilian Society of Immunology. And also the prize for women in science in 2018 that is awarded by L'Oréal, UNESCO, and the Brazilian Academy of Science. She was an assistant professor at the Department of Biochemistry and Immunology at the Federal University of Minas Gerais. Then last year, she moved to become a researcher at the Inserm-backed Institute for Translational Medicine in liver diseases at the University of Strasbourg in France. She has really great publication records, many citations, really interesting, particularly on this thing that she'll be presenting today on the role of microbiota in infectious diseases. So, Angelica, once again, thank you very much for accepting our invitation to speak at the Immuno webinars, and you have the floor now.
Thanks so much, Bruno, for the invitation and to be here for this very nice Immune webinar to discuss. Let's just try share here. Okay. So everyone can see? It's okay?
Yes, it's working fine.
Oh, okay, thank you. So I will start talking a little bit about what I have been developing regarding to the importance of the gut microbiota on infection disease. And I hope that until the end of this webinar, I can convince you how important it is to use the gut microbiota as a strategy to prevent or even control infection disease. So I like starting saying that, as Hippocrates already have been saying, that all the disease begins in the gut. And it's not far for us today that the gut microbiota has been recognized as an important key play in human health, but also in disease. So that's why we know that the gut microbiota are involved in several different diseases, even going through metabolic disease, inflammatory disease, and even cancer. So especially in cancer today, they are very concerned on how we can use the microbiota to treat, especially using immunotherapy therapies to treat cancer. And what I want to go focus on today is on how this gut microbiota could play a role on the host physiology and how this can be important for infection disease. One important aspect that I want to highlight for you about the microbiota is how the microbiota interact with the host. So we know that the microbiota, as a population, the diversity of this population, it's really important, and it's really important also to develop our immune system. So we know that different species of microorganisms in our gut microbiota can play different roles on the differentiation, for example, of different types of immune cells. But they also, it's they also are very important to control the metabolism of the host. I think this is one of the part of the microbiota that I want to highlight here, that I'm working more on this. But if we think about the importance of our microbiota to the host, they are so important on the functionality of this microbiota is so important for the host as well, or metabolic organ that is delivered. So it's well known that to keep the balance of this microorganism composition, it's important to promote a symbiotic relationship with the host. So when we talk about this symbiotic relationship, we know that several metabolites that are produced by commensal bacteria, for example, short-chain fatty acid that I will talk about here on this presentation, they are so important to control the immunoregulation, to keep homeostasis, host homeostasis. But in the other hand, we know that if factors can contribute to the disbalance of this composition, or even by reducing the diversity of this microorganism, we can induce a situation that is characterized as dysbiosis. And this dysbiosis is really impactful for the host, because we can, with this disequilibrium, we can induce that many microorganisms that we consider like pathobionts, that is normal condition, they will not do anything against the host. But when we have this dysbiosis effect, they can become pathogenic. And then they can induce disease or even induce infection on this host. So the dysbiosis situation on the microbiota, they promote immunodysregulation, and then especially in the gut, where we have the most predominant population of microorganisms, they can induce chronic inflammation, and then can impact the host systemically. But when we talk about the strategy or the factors that could contribute with this balance, we know also that different factors might have effect on the gut microbiota composition. The genetic of the host, and even the type of born, if the maternal transfer, or that is really important for the early colonization, if it's like a normal birth or if it's cesarean. The use of antibiotics or even other medications can also impact the microbial composition. Infection disease can also induce alteration of this composition. Many inflammation, many disease, and also stress, and even the hypothesis of hygiene is really true when we talk about alteration on the microbial composition, and also the age. But one of the major aspects that can be impact the microbial composition is the diet. So we know already that if we have like a diet that is rich in fiber, for example, they can promote important equilibrium and keep the balance of this microbial composition that is beneficial for the host. And I will go through this on this presentation. But it's well known also that one type of diet that is rich in lipids and poor on fiber, for example, characterized, for example, by the Western diets, they can induce alteration of the microbial composition, and then this can be one of the important factors that are related to development of obesity and other chronic disease, for example. So as I mentioned to you, I have been shown that when we fed, for example, mice with a high fiber diet, we could see that this diet, they can promote alteration on the gut microbiota functionality by increase the levels of short-chain fatty acids. And here we can see that by 16S sequencing, that when we fed mice with a high fiber diet and we compare with mice with a normal fiber diet, just take the point here. With the normal fiber diet, with the fiber diet, it's a problem here. With the normal fiber diet compared with the high fiber diet, we cannot see much difference on the composition when we look at just by the 16S sequencing of this. But in the other hand, when we look in the right screen, when we look for the production of short-chain fatty acids, especially acetate, that's the most abundant short-chain fatty acid produced by commensal bacteria, we could see that there is a high levels of short-chain fatty acid, of acetate, in the systemic levels of this acetate. So here we use a fiber that is a soluble fiber, that's a pectin, that is a type of fiber that is rich on citric fruit and also in many other type of foods that is beneficial for the microbiota. So suggests us that how we that even that if the composition do not change much, we are able to use diet to change the levels of metabolites produced by the gut microbiota. But what we look at, and I have been exploring this idea that a high fiber diet is important to promote the levels, to increase the levels of short-chain fatty acids. We have been shown that when we fed mice with a high fiber diet, the hypothesis that we have is that when this fiber have been metabolized or fermented by bacteria, commensal bacteria that is already known to be beneficial for the host, and then ferment this fiber to produce the short-chain fatty acids. The short-chain fatty acids have been considering to be very important to promote gut homeostasis because they serve as a source of energy for the colonocytes. We have been shown that when we fed mice with this fiber diet and we promote injury in the gut of these mice by induce colitis with DSS, this dextran sulfate that promote injury in the epithelial cells, we could protect the mice of the injury by just giving them the high fiber diet. And what we found on that time, that one of the mechanisms that are involved on this protection is the ability of the high fiber diet. They activate inflammation pathways on epithelial cells that promote the epithelial barrier, keep the epithelial barrier, and so keeping the epithelial integrity in the gut, and then this protect the mice from the colitis. We have been explore how this mechanism could be involved on that, and at that time, with Charles McKay in Australia and Mauro Teixeira during my PhD, they have the group of Charles McKay in Australia. They have been found that mice, GPR43 knockout mice, they had susceptibility. They have more susceptibility to colitis. When we induce colitis on these mice. So the GPR43 is a G-copoprotein receptor that recognizes specifically the acetate that is microbial metabolite. So we fed the GPR43 knockout mice with a high fiber diet, and this protective effect that we observe with the high fiber diet, they just disappear. And one of the mechanisms that we found there is that exactly the fact that on the epithelial cells, on the gut epithelial cells of these knockout mice, we do not see activation of inflammation on epithelial cell. And we know from that time, from the other labs, that activation of inflammation on epithelial cells, it's really important to keep the epithelial regeneration. And that was one of the idea that why we could not see this protection with high fiber diets. So what I'm telling here is that in the absence of the activation of the GPR43, we do not see the protective effect of diet, of high fiber diet on colitis. And that time, this was the first work that provide the mechanism link that how diet could promote beneficial effect for the host, especially on this intestinal inflammation, through activation of this receptor, that's the GPR43 receptor. To prove that the effect of high fiber diet on the epithelial integrity is dependent on the microbiota activity, We did a simple experiment of fecal microbiota transplantation of feces from mice that receive diet rich in fiber, compare with mice that receive a normal diet with normal composition of fiber in the diet. And we transfer the feces of these donors to the germ-free mice. So we recognize the germ-free mice with this fecal transplantation from these two different groups. And what we observed that mice, the germ-free mice that receive the feces from the donors that had been fed on high fiber diet, they also present activation of inflammation by activity of caspase-1 on the epithelial cells. And I did not show here, but to make the story simple, but they also present reduced inflammatory condition and more and higher survival on colitis. So again, showing that the beneficial effect that we saw from the high fiber diet is on microbiota dependent. And I'm talking about the mechanism that we found that it's this effect at least is dependent on the GPR43 activation, but we know that there is several type of foods that can promote different metabolites that could be produced by the gut microbiota that can bind to different receptors that can also express differently on different type of cells, so with different mechanism and activity. So it's really complex what we are talking about here, because we know that different kind of food can promote different alterations on the gut microbiota functionality that might promote different actions depend on the type of receptor, the type of mechanism, and also what is the major effect that we are expect when we consider the immunity whole of that. So it's really complex, but we are still trying to understand more and more about this mechanism. But as I mentioned to you, one of the aspect that took my attention is that although these metabolites, they are produced in the gut by commensal bacteria that are in the gut, they reach high levels systemically, and the short-chain fatty acids, especially acetate, they go in high levels in the blood, so they can reach different parts of the blood. So they might have different effects on different parts in the gut. And this was exactly what we found, is that when we explore the expression of, for example, of the GPR43 receptor in the inflammatory cells, they are very high expressed on inflammatory cells such as macrophage, neutrophils, and eosinophils. So that's why we become more interesting to explore what could be the effect of these metabolites in other conditions, in other inflammatory conditions. And to start exploring this mechanism, we use a model of gout. That is a model, it's a gout arthritis. That's a model where we induce inflammation in the knee of mice by inject a crystal of urate monocyte. And this promotes recruitment of neutrophils to the knee cavity of the mice. And what we found on that time, that when we treated orally mice with acetate, for example, we could see that the treatment with acetate, they reduce the recruitment of neutrophils, they reduce the presence of neutrophils in the knee after 15 hours, as we can see here in the graph. But one of the results that took our attention here is that they do not inhibit the infiltration of neutrophils in the knee. They reduce the presence of neutrophils there, which suggests us that this treatment promote a resolutive effect by inducing apoptosis of these neutrophils, so reduce the survival of these cells on the inflammatory site. And if they also promote pro-resolutive mediators, we check for the annexin 1, that is a pro-resolutive mediator. And in the time when we see that in both non-treated and treated mice, we have the same amount of neutrophils. And then we observed that the neutrophils from mice that are treated with acetate, they had high levels of high expression of annexin 1 on that. And when we count the efferocytosis of these apoptotic neutrophils, we could see that the macrophages that are in the seat of inflammation, they can phagocyte better this apoptotic body of neutrophils, so increasing the efferocytosis. So all these mechanisms that we observe is a mechanism that suggests that this metabolite, they promote resolution of inflammation instead of inhibit inflammation. And it's for that idea that we start to be more interesting on study the perspective of studying infection disease, because it's well known that when we have an infection disease, we need the inflammatory response to deal with the pathogen, but we also need to control the inflammation to resolve this inflammatory response and to avoid the chronicity of this inflammation. So then we start thinking about how we could use the gut microbiota through production of these metabolites to control infection disease. And on that time, we starting also to explore the gut-lung axis. So the first idea that we decide to look at is what could be the effect of these metabolites on this axis. And we have known from the data from the lab that I have been working with Mauro Teixeira's lab, that germ-free mice, for example, when we infected these mice with Klebsiella pneumoniae that induce pneumonia on these mice, the germ-free mice, they are much more susceptible to the disease. So they die very fast when we infect it with the bacteria. And when we see the amount of CFU on the lung of these mice, there is high levels of CFU of this bacteria in the lung of germ-free mice. So suggest that the germ-free mice, they do not have one proper immune response against the bacteria, and that's probably the reason why they die and they succumb to the disease. So for us, what was interesting is that when we infected the GPR43 knockout mice, for example, they present the phenotype very similar to germ-free. So in the absence of the GPR43 receptor, these mice, they are more susceptible to the they are more susceptible to the infection, to the lung infection. And when we look at the CFU of the bacteria in the lung, they are high increase on the lung of the GPR42 knockout mice. And one of the mechanisms that are related to this phenotype is the deficiency of macrophage, alveolar macrophage, to phagocyte the bacteria. So that show us that the absence of GPR43 activation, that's the receptor that recognizes this metabolite produced by the gut microbiota, we have reduction of immune response to deal with this infection. So, in the other hand, when we colonize germ-free mice with bacteria that we know that are very well producer of acetate, of the short-chain fatty acids, when we colonize this germ-free with this bacteria in live, we could see that the germ-free mice restore the ability to deal with the infection against Klebsiella pneumoniae. And even that when we treated these mice with the bacteria that is inactivate like that, we see some protection, but it's not strong as when we give this bacteria live. And when we look for the amount of bacteria, the bifidobacteria among the Klebsiella pneumoniae in the lung, when we treated with the bacteria, Bifidobacterium longo live, we saw that there is almost no CFU in the lung and one reduction of CFU in the germ-free mice that are treated with the inactivated bacteria. This Bifidobacterium long is really interesting because it's a strain that are isolated from health children in Brazil. That belongs to the collection, the life collections of probiotic. Now they are considering as a probiotic because they had already showing had several beneficial effects to the host. To the collection of Professor Flaviano Martins and Jacques Nicoli at the microbiology department at OFMG. So we work together on this project. And just to confirm that some of production of metabolites from this bacteria could have effect on this phenotype, we use acetate orally on white type mice, and then we saw that when we treated the white type mice with acetate by oral treatment, we could see that they protect mice from the lethality against the Klebsiella pneumoniae infection, and also reduce the amount of this pathogen on the lung. What is true is that this effect of these metabolites is not only related to the bacterial infection, as we found here, but we also show in collaboration with here in France with François Trottin group, that they have been also shown that the short-chain fatty acid has one important effect on the lung infection by influenza virus, and also in the co-infection of influenza virus with Streptococcus pneumoniae. So it's not only against bacteria, it's not a specific effect to also only to negative bacteria, but also to gram-positive bacteria. So here we found that in mice that are infected with influenza virus, there is a reduction of acetate in systemically. So there is this. They also show that there is a dysbiosis on the gut microbiota and reduction of production of short-chain fatty acids. And when we treated with acetate mice infected with co-infection of influenza virus and Streptococcus pneumoniae, the treatment with acetate prevent the lethality of these mice, and also they reduce the production of acetate, they restore the production of acetate in the blood, and reduce the amount of CFU in these mice. So it's not, again, it's not a specific effect to bacteria. So when during this time that we are just exploring the effect of the gut microbiota and the gut-lung axis, the COVID pandemic arrived. So considering all the strategies that were at that time using to treat COVID, we are very concerned about the use of antibiotics that we know that can impact the gut microbiota, but also the symptoms that some individuals that had COVID present, like diarrhea and other gastro symptoms that could be involved on the alteration of the gut microbiota. So then we become interesting to understand what could be the role of gut microbiota on the COVID disease, especially on the severity of the disease. But in the other hand, this is another story, but in the other hand, we try also to understand what could be the impact of COVID infection on the gut microbiota, and what could be the consequence of this alteration. So there's a lot of work that is starting become at that time, showing that the gut microbiota was altered in patients with COVID. But for us, what not most important to understand who is there, and more important for us was to understand what they are doing. And this direction, what we did, this was performed in my lab, my former lab in Brazil, with Viviane Mendes and Mayra Rich, that is a PhD student at that time and a postdoc in my lab. What we did is that we look at individuals from the same house, so it's co-housing individuals, that one had the COVID and the other one was negative for COVID. And we look for these individuals up to 1 or 4 months after the infection, or after the test that they did that confirmed that they had been infected by COVID. And most of these individuals is volunteers. Most of them, they had just like mild or even asymptomatic symptoms. So when we collect the feces, so we come to this volunteer, we collect the feces, the fresh fecal samples of these individuals. And the first experiment that we did is to analyze the sequence of the microbiota of these individuals. And when we look at the composition of the microbiota, again, we do not see much difference on the composition when we compare controls versus post-COVID individual, that we call them post-COVID since they were not infected at that time. So there is no difference even when you compare beta diversity or alpha diversity, there is no difference on that. And we did not see virus particles in the fecal samples of these individuals. But what took our attention also is that when we look at the production of short-chain fatty acids on the feces of this individual, we observe that even acetate, propionate, and butyrate, they were reduced in this individual that we consider post-COVID individual when we compare with the control. So there is a suggests that there is alteration on the gut microbiota functionality, although we do not see difference on the composition. So what we did is to transfer this microbiota. So we did the fecal microbiota transplantation to germ-free mice in the lab. And then we become very surprised with what we found, because the simple fact of transferring the feces from the germ-free receptor mice, we could see that mice that receive the feces from the post-COVID individual, they present different alterations in different organs. For example, they had more germinal centers in the intestine. They had more inflammation when we look at the lung of these mice. And they also had loss of cognition. They present alteration on behavior in even memory behavior. So all these alteration at that time, no one was talking about the post-COVID sequels. But what we found is that the simple fact of transferring the microbiota from individual that had been exposed to the SARS-CoV-2 virus infection, they had impact on the gut microbiota alteration. And then this alteration resemble the phenotype that we today call them as post-COVID sequel on germ-free mice. Another data that we look at even concern the use of antibiotics is that we look for we isolate these species and then we cultivate them on different mediums in your lab. And what we found is that in individuals, in feces from individuals that have been considered post-COVID individuals, we found that there is a more predominance of multi-resistant and resistant enterobacteria in the feces. So enterobacteria is a group of bacteria that is well known to be characterized as a pathobiont because they are present in high abundance in several, in most all individuals, humans, but they can be pathobiont because in dysbiosis condition they can become pathogenic. And what we look when we isolate individual strains from this group of bacteria, and then we characterize individually, we found that among the enterobacteria that we found to be multi-resistant and resistant, on post-COVID individuals, we have more predominance of, for example, Klebsiella pneumoniae. We found more predominance of bacteria that the WHO consider like a escape group of bacteria, and the name of escape is exactly because they are bacteria of very well concern or today, because they can be they can escape the immune system, and they are very important bacteria that are reservoir of resistant genes. So it's a very well concerned strains for antimicrobial resistance. And we found that Klebsiella, for example, is one of the most important strain that has increased on this post-COVID individual. So to test if this alteration that we observe with this transplantation could impact the physiology of how these mice, for example, could have when we face one infection with a resistant strain, for example. We did the same experimental design. So we collect the feces from individuals from the same house, control versus post-COVID individual, and we did the fecal microbiota transplantation to germ-free mice in the lab. But we divide this group of mice, the receptor mice, in two other groups, and one we infected mice with a strain of Klebsiella pneumoniae that is a multi-resistant strain. And we observed that when we infected the mice with this strain, we observed that mice that received feces from post-COVID individual, they had more inflammatory response in the lung, and even that they had more inflammation by more infiltrate of cells and more cytokine production on the lung. This inflammatory response was not able to deal with the bacteria, because these mice, they still have this bacteria in the lung. So suggest us that the alteration of the gut microbiota could be involved on the deficiency on the host to deal with infection disease. And again, when we look for the short-chain fatty acid concentration on this group of mice, we observe that although we do not see much difference on the acetate levels when we compare the mice that receive feces from donors, the control donors versus the post-COVID donors, when we infect these mice with Klebsiella pneumoniae, the mice that receive feces from the post-COVID individual, they had reduction on the levels of acetate. So again, suggests that the impair of this mechanism against controlling this infection could be related to the reduction of short-chain fatty acids production by the gut microbiota. And one of data that took our attention also is that when we look at this, we come back to these volunteers, the human individuals that we collect all these samples, and we look at the biomarkers for the gut epithelial barrier, for example, for the LPS in the circulation. We do not see much difference, but we saw that there is increased levels of ICB-IBCCF in the serum of these patients, and ICB-IBCCF is considering a biomarker of gut epithelial integrity injury, And then we could see that this individual, they might had alteration on the gut microbiota that could be involved on the epithelial or impair of epithelial integrity. But another aspect that took our attention is that when because we applying a survey to these individuals, and when we look at when we compare the feeding habits that we collect on these clinical surveys, when we compare controls versus post-COVID, we do not see much difference. But in our cohort, we saw that there's only like a small percentage of donors that was considering moderate symptom. And in these donors, we saw that these individuals, they had a very great deficiency on the food that we consider gut microbiota friendly foods, like fiber, probiotics, or many other type of diets and food that could help to promote gut microbiota symbiosis. And this is, we cannot get many conclusions from this data. The only conclusion that we have here is that we don't know if this deficiency of this nutrition status is related with the severity of they present, but it's a data that we become very interested on investigate that. But just to go on this direction, and I'm almost in the end, this highlight us one of idea that we discuss a lot in the lab, that the interplay between diet, microbiota, and host response against infection disease become to that sentence that we use, that we are what we eat. But in this case, what we are trying to see here, that maybe we are what we don't eat. And why I talk about that is because when I go back to this data that I show you at the beginning of the webinar, I highlight here that high fiber versus normal fiber, we do not see much difference on the composition, but we saw that high fiber diet is enough. It's important to increase the production of short-chain fatty acid that's beneficial for the host. But when we look for mice that we just deplete completely the fiber on diet, so we considering then a low fiber diet, these mice, they present a very huge alteration on the gut microbiota. And this is observed even in the composition, in the sequence of that. So to test our hypothesis that we know that, okay, eating fiber is important and it's good for our health, but how is about the deficiency? If we don't eat fiber diary, for example, what could be the impact for the host? And then to explore that, we use a similar experimental design in mice, where we fed mice with a normal fiber and with a diet with depletion completely of fiber. And then we look at the lung of these mice, and what we observe that the simple fact of the mice eating a low fiber diet, they already promote inflammation in the lung. So we have more infiltration of neutrophils in the lung in mice that were fed in a low fiber diet, and with more myeloperoxidase activity of these neutrophils in the lung. And what took our attention also here is that when we infected these mice with bacteria that are with the Klebsiella pneumoniae that are resistant, and we transfer and we look at after the clearance of the bacteria in the lung, and we look at in the gut for bacteria that are resistant to antibiotics, then we observe that 8 days after the clearance of infection, of lung infection, we have predominance of bacteria in the gut that is resistant to antibiotics. So suggests us, and this is only observed when in mice that were fed in a low fiber diet, and not in mice that were fed in a normal diet. So suggests us that the low fiber diet, they can promote alteration on the gut microbiota, and then can promote selection and dissemination of bacteria that are resistant to antibiotic. And just to finalize here, we work on this idea that low fiber diet is one of the characterized by a malnutrition diet that could impact the way that we deal with infection. But we know that other kind of diet, also like high fat diet or even ketogenic diet, they can also affect the way how the host deal with infection. And here I use a model of Drosophila melanogaster, that's the fruit fly, where we fed the Drosophila with different kind of diet, with a standard diet, with high fat diet, and ketogenic diet. Why I'm using high fat diet and ketogenic diet is because we want just to use the different composition of carbohydrate that is important to bacteria that can ferment, that is fermentative. And what we found is that the survival of Klebsiella, of Drosophila melanogaster after exposition of a high fat diet and ketogenic diet, they have more lethality when we infect them with the same strain that we use in mouse, that is the Klebsiella pneumoniae, that is multi-resistant. And we found similar phenotype that when we look at the Enterobacteriaceae group on these mice, with ketogenic diet and high fat diet, we could see increased levels of Enterobacteriaceae on the gut of these Drosophila mice after infection, even that one interesting data here is that even in mice that are in Drosophila that were not infected, the simple fact of feeding them with ketogenic diet, we could see increased levels of Enterobacteriaceae on these mice. So we don't know exactly the mechanism how this could be involved on this responsibility, this lack of responsibility response against the infection, but we are still trying to investigate that. So with that, I just want to the take-home message here is that we might be considering to use the microbiota as a base immunomodulation strategy for infection disease and even to control disease. And again, what is more important is not only look for the who is there, but more important is what they are doing there. And I'm talking about one of the gut metabolites that I explore here, that's the short-chain fatty acid, but for sure there is like several hundred different metabolites that can be produced differently, that can control differently the population, and also the other metabolites that are produced by the other species in the gut. So it's really complex. So we are just open the Pandora box because it's just to increase more complexity on to understand how we could use the microbiota on this. But what is very important here that its diversity matters no matter where, because it's just diversity is not just about how many different microorganisms exist. That's what I show you here, but it's about how they work together to sustain health, resilience, and also innovation. So that's why it's really important for us to considering explore all this diversity of microbiota, but not only the composition, also the metabolites. And this is applied also for the food. We can discuss this in the end, but as I mentioned too, there's several type of nutrients that can promote different aspect and different functionality for the gut microbiota. And then this tell us that how important is the diversity of our nutrition also to deal with this. And then I want just to, oops, I didn't expect that. I just want to finalize here by acknowledgement with all the people that are have been work on this project with me, especially my former lab in Brazil, where we perform most of this experiment, and all the collaborators from the University Federal of Minas Gerais, but also from the Universidade Federal de Rio de Janeiro, Alessandro Filar, that work a lot with us on this lung access, gut lung access, and all the others collaboration, also the University of Strasbourg, where I am now, and all the financial support that I got during this this work. So thank you so much for the presentation and to be here, and I'm open to questions.
All right, Angelica, thank you very much. It was a really, really nice talk. Lots of interesting data and discussion. I'm going to open here with a question from Wendell Laranjeira. That's Laranjeira, singular. That is one of our students here in Rio. Wendell, please.
Good afternoon. I don't know if it's good afternoon where you live, but
Yes.
I want to say first, thank you for your presentation. I really do like your presentation was on point for me, because not many people talk about gut bacteria, gut microbiota, and how important they are for our system. And I think that you put it together in a really good way. Now I'm showing the camera. You put it in a really good way, and I really do like that. I don't know if I'm going to go out of topic right now. Excuse me if I am, but I'm interested in the development part of the gut microbiota. And at some point, you spoke about how different bacteria that are beneficial to us help with the stimuli, infections, and so on. And I've been meaning to ask you this, but do you think, now more in your opinion, gut microbiota is different for, let's say, people that live in Brazil, people that live in India or any other country, because you spoke about the different populations of bacteria. Do you think those populations change, and do this change affect those results that showed us?
Yeah. Definitely, this is a very good and important question, and this go also to the for who is working on the gut microbiota. There is one of concern that we have now is that, and we used to say that because most of what we know about the gut microbiota relationship with the host is basing on the study that is made in bigger population that is in developed countries. And that's why I think, to be honest, I think the most beautiful microbiota that we might be found, it will be in Brazil, because we are very diverse, we are genetically diverse, we are completely different culture, nutrition, it's quite different, but we don't know nothing about our microbiota and how we deal with all these infections because we have so many different infections. So there's a I would say that there's a huge of people that is starting concern that we should look more for the relationship of gut microbiota in Brazil and even in other countries like in Africa also to see how they behave because they are always exposed to different infection, different disease, different conditions. And one important thing about the gut microbiota because they are very resilient. So even though we change the equilibrium of the microbiota many times, especially with diet. They are very resilient to what we are. So that's why it's really important to considering that. So this is one very important point. But for sure, the microbiota for individuals from different place in the world are completely different, even that they are not completely different, like the signature that what we found is not completely different, the way they behave is completely different. So that's why we need more and more study to explore this behavior. So
All
thanks
right, thank you.
for your question.
Right, thank you. We don't have any further questions from students here. I don't know if I have questions from students in São Paulo or Uberlândia or elsewhere, people that are online.
So, Bruno, we don't have questions from students here. If there's no questions in the chat, I have a question.
Okay, so why don't you go ahead, Diego?
Okay, so very nice talk, Angelica. Thank you very much for sharing your data. I was curious about an aspect, but for example, in that experiment where you see that for the post-COVID patients and the associations with that, where you see this infiltration of neutrophils when you colonize the mice prior to infection, right? My question is, whether do you believe it's just the neutrophils that are mediating that higher susceptibility, or what would be the role of the lung microbiota that may be changed because of that?
Yeah, no, that's the question that we cannot give you now because for sure from the post-COVID experiment, we know that the microbiota on the lung is different. So again, we don't know. We know for sure that even the colonization of the microbiota on these mice is different, and that it's what bring or the concern about how could be the alteration of this gut microbiota to the secondary infection, because the idea at the beginning is exactly that we are changing this microbiota in the post-COVID individuals, and we are favor bacteria that are more resistant. And we know that these bacteria are also more able to disseminate and go to the other tissue and organs. So that we do not explore which kind of bacteria are in the lung and what could be the whole of that. But for sure, they are different. But this is also true for the experiment that we did with the diet. So even when we fed mice with different diet, The lung microbiota is completely different. So that's why we cannot exclude that the effect is also the alteration on the lung microbiota, and which is the more important that are playing a role on the, at least on the lung inflammation. So I don't think that it's just neutrophils that is playing the role. I know that they are there because they are there. They are playing a role because we look for them. But of course, there's other mechanisms that could be involved. And I think Denise Fonseca also is looking at other immune cells that when, for example, when we fed with a ketogenic diet, there is more also neutrophil, but there is more infiltration of IFCs, if I'm not wrong, in the lung. But again, we don't know if all this immune alteration that we see in the lung is related to the alteration on the lung microbiota. So we cannot exclude.
All right, thank you.
All right, so I have a question of my own, and then Miriam has a question as well. But I was wondering, it comes from a place of really ignorance on my part. Is it's very clear that high-fat diet is very important for our health. But I was wondering how plastic is the microbiota, and how long it takes for it to change for a healthier microbiota that will actually make us benefit from these diets. And also being, I'd say, someone that was with a patient in a hospital, it doesn't seem that hospital diet is all that rich in fiber, is particularly rich in green jello. But what's we are missing that we are not applying these kind of diets in our hospitals as well, not just on our day-to-day lives.
Yeah, so two very important points that you raise here. One is that there is one paper, but again, in showing mice, because it's very complicated to follow up individuals with this kind of diet, but is starting. There is one paper showing that, so with one day, if you change your diet to a Western diet, for example, with a diet with a poor nutrition, for example, it's able to change your diet, your microbiota in one day. It's enough to change. But as I mentioned to you, the resilience of the microbiota is really high. So even that you change one day of your diet, for example, if you go to the fast food one day and you eat, you will for sure change your microbiota. But the microbiota will try to restore what they were, because the pressure of the host interaction with them is more important than this like a short exposition of diet. But in the other hand, they have been showing that people that, or even mice that are fed on the high fiber diet or in diet that's really rich on these nutrients for commensal bacteria, the alteration of the gut microbiota seems to be more prolonged. So you change, but the dysbiosis or the unbalance induced by this malnutrition diet, it's compensate if you have a diet that is more beneficial for the gut microbiota. So again, I mean, this give us hope that when we have like a health diet, like a for long regime of diet, it's more and more beneficial even to control this alteration, this dysbiotic alteration that you might have when we change very quickly the diet. But one important thing about this is that the window for development of this good microbiota to promote this health resilience, it's really important at the beginning when the microbiota are developing. That is when the 3 years after you're born, the birth. So that's why it's important to considering that. And what is the second point again?
In hospitals, why hospitals are not applying this kind of diet.
This is something that, for example, it's how science and reality is completely crazy. The translation of these things is really complicated because we discussed a lot with nutrition at that time because even like a reduction of food maps that is most rich in fiber, and in your hospital they take out the fiber exactly because they cannot promote a transient colon effect. So we discussed that why they do that, because we are seeing different, because they are just considering the physiology of how fiber we're considering, and not taking consideration that how fiber is important for the immune system and also for that. But then now I think they start to change that. So it's not like a rule today to considering that high fiber diet should be included in that, but at least they are considering to use a supplementary of soluble fiber, because the physiological effect of soluble versus insoluble fiber is different. And we know also that soluble fiber is one of the substrate that's more important to production of short-chain fatty acids, for example. But the other idea could be also using the short-chain fatty acids during this period instead of using fiber. And you have also to consider that I didn't mention here that we are talking about a system that depends on diet, microbiota, and the production of short-chain fatty acids and the host genetic. Because what we think is that even that you give plant of fiber for someone that there is no commensal bacteria that are producer or ferment this fiber, there is no effect. And if the individual has some, I don't know, deficiency on the expression of this receptor, for example, the GPR receptor, they might be not respond to the beneficial effect that we are seeing. So that's why it's again complicated. It's not quite simple to go for this.
Sure. Thank you very much. We have a question here from media now.
Hi, Angelica. Thank you very much for your talk, and for accepting the invitation. It's really stimulating indeed. I have a couple of questions that go along with Bruno's and with Diego's questions. So just to continue on Bruno's discussion, my question was, for those people who don't eat well, right? I think about my kids, for instance, and we think, oh, let me give him a probiotic to see if I can compensate, but they don't eat the fiber to stimulate that bacteria to stay along. Do you have any studies on that? Like how does it make any difference?
No, make difference for sure, because again, one of the fiber is only to stimulate the production of the short-chain fatty acid, right? To elevate the levels, but doesn't mean that the production of even the basal production of these metabolites are not important. So that's why I always defend that it's important to give probiotic. I'm defend a lot now the fecal microbiota transplantation for sure, because I think this is the future. But the problem is we don't know exactly how is the interaction of these microorganisms with the host. I mean, we are. That's why, for example, why the probiotic, because probiotic exists for 100 years, but why they are not they do not work well for all the individuals, because it's really personalized. So that's why we need to look more individually for the microbiota, for the functionality, for the lifestyle of the individual, for where they are, to modulate, to use the microbiota as a really therapy. So this is what we defend since the beginning. But as I mentioned on the beginning of the webinar, what I think now it's really take serious that microbiota is something important is again when we are looking for the immunotherapy of cancer, because I don't know if you are aware of that, but when they starting doing the immunotherapy to individual that they starting respond and non-responder to the immunotherapy, and this is not only for CAR cells but also for anti-PD-1 and anti-PD-L1 in cancer, they realize that individuals that do not respond, they look at why they could not be respond to the immunotherapy. And when we look at the microbiota are completely different. And okay, the microbiota are different, but what they decide is that if we do the fecal microbiota transplantation for the individual that respond to the non-responder, they become responders to the immunotherapy. And yes, that's exactly what happened. So because immunotherapy is really expensive also, now the clinical trial is starting considering the microbiota as an important player on this immunotherapy. And now I think they will start considering the microbiota as a personal biomarker to analyze, to go through in detail, and not only cancer, but all other disease. So I think it's really necessary to see individually.
No,
So
that's really interesting. And so now touching on Diego's question, I was wondering, I saw some work back in the day that you see circulating bacteria that don't elicit sepsis, right? You do find them. And so what makes me think that you have exchange between, let's say, lung and gut microbiota naturally. So have you done an experiment? So do you see if you can replenish gut bacteria, pulmonary microbiota by doing also those transplants, or do you think about that when you're doing the experiments?
we think about, but as I mentioned, it's really complicated because one thing that we know, for example, if you colonize only the lung, if you do a microbiota, for example, in the lung, then we will see that there is colonization in the gut. So what we know is that we are not sure how this can drive this colonization, but we already know that oral bacteria and oral microbiota and the gut microbiota is the major drive to to induce decolonization in the other tissues and to drive the successive colonization in the right direction. How we can interfere on that is really, really difficult. So, but for sure, there is a lot of things that we don't know. And another thing that I think is important to mention here is that we still deal with how to look at this microbiota, especially, for example, the microbial components that are circulating. It's really difficult to detect this in high quantity, and so it's a lot of technical issues that is still ongoing to solve, because with the tumor microenvironment is the same. We know there is some microbiota composition inside the tumor, especially in solid tumor, but we don't know exactly how to identify as a signature. So there is a lot of the data from different places that are very confused because of this contamination, and there are several things. And also there is also other mechanisms that how the microbiota can be impacted, that's like for the exosome production that microbiota can produce, and also for these metabolites, so, and for sure the metabolites. So it's a lot of way that the microbiota can impact colonization, immune system in different sites of organ that is not only in the gut.
That's amazing. It's another organ, right? It's like an endocrine.
Complete.
So, Julio, there is a question by Julio Schafferstein on the chat. Julio, would you like to open your video, or would you prefer us to read the question for you? I see that he's still logged in, but if...
Você está me ouvindo? Está me ouvindo?
Yes, yes.
Tô ouvindo.
Que legal. Peraí, deixa eu botar o vídeo também, que assim eu vejo. A Angélica pode me ver. Eu não sei se está reagindo o vídeo. Ah, agora sim. Está me vendo?
Não, só.
Não.
Não
Eu
apareceu, não.
estava olhando o outro computador. Apareceu? Não. Bom, esquece então isso, tá? Deixa eu... tá desligado. Então, a minha pergunta é mais relacionada com essa parte básica que você começou, né? Porque a apresentação é uma apresentação muito ampla, cobre tantos assuntos fascinantes. Nossa, eu queria só ver se você sabe alguma coisa a mais relacionada com esses estímulos para induzir a apoptose de neutrófilos. e saber, entende, se você acha que isso é exclusivo dos neutrófilos ou existe uma network muito mais ampla, né? Porque você não sabe. Você fala de resolução da inflamação, talvez você possa ter os mastócitos, eu não sei como é que estão a própria célula endotelial, não tenho conhecimento se a GP43 é expressa, se ela responde ao acetato, né? Que essa interação toda com o neutrófilo começa sempre com endotélio. Claro, pode envolver plaqueta, não tem fim essas coisas.
Não.
Como é que você imagina, né? Porque nesse quadro que você desenhou, ficou realmente assim um buraco, né, de conhecimento, né?
É. Não, Júlio, assim, concordo plenamente. Realmente esse buraco existe, até mesmo porque a gente ainda não entende muito bem da fisiologia do receptor. Mas uma coisa que eu posso acrescentar de complexidade é que o efeito dos ácidos graxos de cadeia curta, principalmente nessa questão de imunomodulação, não é somente dependente do receptor, porque eles são ácidos graxos, então eles podem ter efeitos,
Claro.
entram direto na célula. O GPR43, por exemplo, ele é altamente expresso, né? Na época que a gente viu onde ele está mais expresso, que é nas células inflamatórias e nas células epiteliais do intestino, mas com single cell hoje a gente está vendo que, na verdade, ele tem
Muito amplo, né? Os
muito amplo,
macrófagos
muito
também
amplo.
são muito
É,
sensíveis. É.
então a gente não sabe muito ainda do efeito, assim, principalmente nessa questão da resolução. Realmente, na época, a gente focou no neutrófilo, porque era a nossa célula, os modelos que a gente estudava, era neutrófilo eram as células principais, a gente focou muito nisso. Mas sem dúvida, assim, que tem outros efeitos que podem estar envolvidos,
Relacionados com isso, né?
relacionados a
É,
isso, com certeza. É, então, assim, isso é um mundo ainda a ser investigado, é onde eu
é. Eu
pretendo.
fiquei curioso, porque eu tive um contato muito recente, semana passada, com um grupo na Suécia que trabalha com mastócitos e estuda a apoptose de mastócitos. Eu nunca tinha pensado sequer sobre isso, né? E aí, quando você mencionou esse efeito, e aí você vai, entra dentro do pulmão, né? Eu pensei, puxa, pode ser que essas coisas tenham realmente uma relação com o teu modelo também, né? Uma
Não, com
influência.
certeza, mas assim, sinceramente, a gente não chegou a olhar mastócito, então vou falar que eu não sei porque realmente também a gente não olhou, e eu nem sei se dos dados na época de microarray que a gente fez, se como é que era a expressão de GPR53, por exemplo, em mastócito. A gente pode até voltar nos dados, porque a gente focou nas células inflamatórias que estavam mais envolvidas nos modelos que a gente estava estudando. Mas
Claro,
assim, é muito essa que é a parte bonita, assim, porque tem muita coisa que esses metabólitos agem indiretamente também, né? Assim, sem ser um efeito direto. Porque, por
claro.
exemplo, o butirato, ele é muito bem conhecido de de inibir estona deacetilase, então, muito dos efeitos anti-inflamatórios que ele pode exercer em vários contextos é pela inibição de estona deacetilase, e por isso inibir a expressão de alguns genes inflamatórios, mas, enfim, eu acho que é um caminho ainda muito
É.
para ser
Bom,
percorrido.
como todo mundo, estou agradecendo, né? A gente aprendeu muito com você e realmente só o trabalho também de Covid já dava uma outra palestra e aí mundo afora. Muito obrigado, foi um
Eu
prazer
que
enorme
agradeço,
te ouvir.
Júlio. Obrigada.
Thank you, Julio. So Maria also has a question.
Oi, Angélica, obrigada de novo por ter aceito o convite e pela belíssima conferência que você nos deu. Muito legal, um trabalho muito bonito.
Obrigada.
Então, aí eu também vou puxar a brasa para as minhas células favoritas, né? Aí eu vou perguntar das células T. E porque assim, a gente sabe, né, o butirato em célula Treg, né, é super importante. Mas eu, em particular, quando você falou em pulmão, na infecção do pulmão, eu pensei nas made T cells, né, aquelas células que estão ali na fronteira entre imunidade ah, adquirida e adaptativa, que são células residentes do pulmão e que, né, aí eu fiquei pensando, será que tem alguma coisa? Elas
Olha,
tem, expressam esses receptores também, também.
não, linfócito T regulador, expressam muito a GPR43, mas, de novo, não é. Tem vários trabalhos, inclusive considerando o papel dos ácidos graxos, né? Do acetato, do propionato também. E uma coisa que eu não falei é porque tanto o propionato quanto o butirato, por exemplo, eles também se ligam em outros receptores, né? Tem ações diferentes e vias inflamatórias diferentes, atingem vias inflamatórias diferentes. Por exemplo, o GPR-109, então aí já são outras cascatas de mecanismos que podem atuar. E com o linfócito, tem muitos trabalhos já mostrando como que esses ácidos graxos de cadeia curta, eles acabam modulando os linfócitos T e levando a uma resposta até mais reguladora e etc. Mas no pulmão, outros tipos celulares eu não olhei. É, assim,
Eu estava pensando.
como eu acabei vindo para a França, e aqui, na verdade, a gente ia focar mais na parte do gut-liver axis, né? Que aí também é onde eu fiquei mais interessada em saber como que esses metabólitos alteraria a função metabólica dessas células, porque eles são metabólitos que acabam alterando de uma maneira o metabolismo celular, e é isso que mudaria um fenótipo que a gente poderia estar vendo. Então, assim, é, eu não segui, não dei sequência mais em explorar mais aprofundadamente outros tipos celulares no pulmão e quais seriam os outros papéis que esses ácidos graxos poderiam estar lá. Realmente eu foquei muito no macrófago, alveolar e no neutrófilo, mas eu posso até checar com a Alessandra. Ah, não, mentira, porque a Alessandra Filati teve uma época que ela chegou a fazer um screening pra gente do que estava acontecendo, mas foi somente com as dietas. Nós não chegamos a olhar, por exemplo, com os metabólitos em si diretamente, o que estaria acontecendo com essas outras células que a gente não focou. Mas não,
Obrigada.
não ficaria surpresa com outros mecanismos que estariam envolvidos, Maria.
É, porque em célula T, como você disse, né, eu sei que tem muita coisa, mas especificamente nas mate T cells do pulmão, que são muito importantes no pulmão, isso eu não, enfim, não sei, não sigo essa literatura, fiquei curiosa, mas de qualquer forma, parece uma coisa interessante de ser feita, mas obrigada de novo, é tanta coisa, são tantas perguntas, muito bonito, obrigada.
Eu que agradeço, Maria. Eu acho que não tem mais, né?
I'm
Nós temos
sorry.
uma pergunta de
We have
uma aluna.
another question from São Paulo.
Ah,
Uma pergunta de uma aluna aqui.
Angélica, obrigada pela sua apresentação. A minha questão ela é mais inicial, do início da apresentação. Eu vi que
uhum.
vocês usaram um modelo de gota e vocês avaliaram, né, o infiltrado neutrofílico. A minha dúvida é se vocês chegaram a avaliar o inflamassoma, porque eu trabalho com inflamassoma e aí isso me veio a dúvida.
Não, então. Aliás, esse é um dado até bem interessante para a gente, que no início foi meio contraditório, porque no intestino, nas células epiteliais, a gente via que a ausência do receptor, do GPR43, no nocaute, a gente tem deficiência da ativação de inflamassoma nas células epiteliais. Quando a gente foi fazer gota, a gente esperava exatamente o fenótipo oposto, porque, tá, a gente não vai desenvolver gota. Se a gente tratar com acetato, a gente não vai desenvolver gota nesses animais, porque ele não vai ativar inflamassoma. E o que nos surpreendeu é que, de fato, e a gente tem esse trabalho, inclusive, que a gente fez primeiro no germe-free. Então, na ausência da microbiota, como é que era a gota no germe-free? E, de fato, o germe-free não desenvolve gota. E ele não desenvolve gota, ou seja, ele não tem recrutamento de neutrófilo, o germe, na ausência de microbiota, você não tem ativação de inflamassoma também no modelo de gota. Só que quando a gente dá acetato para o germe free, aí ele restaura. Ou seja, ele passa a ativar a inflamassoma e ele passa a recrutar o neutrófilo. Só que ele é como se ele primasse, por ativar o complexo de inflamassoma, ele passa a responder ao estímulo da gota, mas não a gente também não tem como comparar, porque o modelo de gota que a gente usa, né, como o camundongo é autorresolutivo, né, porque ele tem a urease, então a gente não conseguiu cronificar para saber se o quanto que esse estímulo que a gente estava dando com acetato no germe-free poderia prolongar. Mas quando a gente compara com o animal WT, né, convencional, ele restaura o fenótipo, mas ele ainda reduzido assim, comparado com o convencional. Então, assim, o que a gente viu é que, de fato, acetato estimula a ativação de inflamassoma, é via ROS, se a gente usa o inibidor de ROS, então a gente acaba não tendo efeito, né, quando a gente trata com acetato. E aí, quando a gente foi dar o acetato, então, para o animal convencional, a gente pensou, pronto, agora o animal vai ficar pior, então realmente é um mecanismo pró-inflamatório que esse metabólito induz. Mas não, o que surpreendeu a gente foi exatamente isso, assim, né, até pela cinética, que na verdade a inflamação ela não é inibida e nem é maior. O que acontece é que num determinado momento você tem o swift da resposta inflamatória exatamente para mecanismos pró-resolutivos, que foi aonde exatamente a gente foi investigar, então, se tinha mais produção de mediadores pró-resolutivos que justificassem por que dessa redução, né, antecipada do neutrófilo na cavidade. Então, assim, tem ativação do inflamassoma, o estímulo que os metabólitos fazem é ativar a inflamassoma, isso parece ser dependente da ativação do GPR43, mas de alguma forma, esse é o buraco que a gente tem, é como que esse controle de uma resposta exacerbada é controlada por esses metabólitos a direcionar para uma pró-resolução. E eu acho que, assim, de uma maneira mais geral, é isso que do ponto de vista mais romântico da microbiota é exatamente o papel da microbiota, né? É tentar ensinar o hospedeiro a como responder a um estímulo inflamatório e a lidar com aquele estímulo inflamatório para homeostasia, né? Que é exatamente o que a microbiota faz no intestino, né? Para ela se colonizar lá, ela precisa de estimular uma resposta inflamatória, mas que também estimula respostas que são imunorreguladoras para controlar essa população. Então, bom, isso também é sistêmico, pelo visto.
Muito obrigada.
De nada.
All right. So thank you very much. I believe that was our last question. We would like to thank once again for Angelica for this excellent talk. Very exciting. We actually had one comment in the chat about clinical applications, but I believe that we covered that. And but for now, this is our last regularly scheduled presentation of this year. So I'd like to thank everybody, my co-organizers in this endeavor. So Gustavo, Diego, Tiago, Miriam, Cristiane, we are very happy for the success of one more year of immunowebinars. We are hoping to see everybody again next year. But first, we are going to have this extra presentation in 2 weeks. That I would like to ask Diego to talk a little bit more about this next presentation, please.
Yeah, so we're going to have this presentation by Guilherme Nader from University of Pennsylvania. So he's going to be around. He works a lot on imaging, So he's going to be talking about that with us and bringing his project. So everybody who wants to join, we're going to do the transmission online too, and we're going to share the link. So soon I will be sharing the folder for his talk too, and we look forward to seeing all of you again.
All right, great. So thank you very much, everybody. Have wonderful holidays, and we'll see everybody either in 2 weeks or next year. All right, thank you. Bye-bye.
Tchau, tchau, pessoal. Até mais.
Tchau, tchau.
Ô, Bruno, poderia ter pedido para ele mudar o microfone, né?